Hi, I'm Lyndsay Millican, and my thesis proposal focuses on finding a cure for TMJ disorders. So what is TMJ exactly? TMJ stands for the temporomandibular joint, which is located between the jaw and the cranium. More specifically, TMJ lies between the mandibular bone of the jaw and the glenoid fossa of the temporal bone of the cranium. The TMJ is a load-bearing synovial joint disc that aids in primal functions such as chewing and speaking. Zooming in on the TMJ region, you can see the glenoid fossa of the temporal bone here and the mandibular condyle here. Within these two bones lies the actual temporomandibular joint disc. The disc itself is an articular disc that separates the joint into superior and inferior components. The articular disc is deficient of blood vessels and nerves and consists of fibrocartilage, whereas other load-bearing joints of the skeleton consist of hyaline cartilage. The TMJ disc is composed of different cell types that include synovial cells, disc cells, and chondrocytes that have different cell morphologies and functions. These cells are surrounded by their extracellular matrix, which is comprised mainly of collagen, which plays a significant role in structure and cell signaling for growth, division, and even death via apoptosis. The ECM also contains proteoglycans, which are proteins with sugar chains attached, and specifically lubricin, or proteoglycan 4, which aids in lubricating the TMJ region. Versicon is also a key proteoglycan that is involved in cell adhesion, growth, and migration. What are TMJ disorders? TMJ disorders affect a large portion of the U.S. population, with the majority being women of childbearing age. Symptoms of TMJ disorders include lockjaw, limited mouth opening, clicking, dislocation, and pain during jaw movement. The cause of TMJ disorders is not well understood. Some hypotheses suggest the disorder could be due to mechanical stress, genetic causes, or hormone differences, such as much greater number of females reporting TMJ disorders than males. My research will focus on testing different extracellular matrix molecules of TMJ disc cells for protein breakdown. The TMJ disc cells were isolated from baboons in the San Antonio area, chosen because they have a very similar TMJ environment to that of humans. The research analyzes the central dogma of biology, in that cells convert DNA to RNA via transcription and then RNA to protein via translation. Previous studies demonstrated that the promoter activity of mRNA expression of the lubricin gene, which is also known as proteoglycan 4, is downregulated in the presence of estrogen. In other words, there is evidence that TMJ cells transcribe the lubricin gene into mRNA and could be a potential reason behind the cause of the gender discrepancy of TMJ disorders. However, translation to protein evidence is needed in order to determine if disc cells are able to make and secrete the lubricin gene into the extracellular matrix. Not all genes encode for proteins, as some RNA fragments are spliced out as introns and removed from the mRNA sequence prior to translation. It is therefore hypothesized that quantification and protein breakdown of the extracellular matrix molecules lubricin, big H3, versicon, and collagen types 1 and 2, synthesized by TMJ disc cells, should indicate that the cells translate the mRNA genes into protein and secrete the desired proteins into the matrix. Methods used to quantify protein breakdown will include Western blotting and densitometry. The results of this study will help characterize the TMJ male and female disc cell lines and provide more information for future studies. If the central dogma for TMJ cells is better understood, this will provide a basis for how to treat TMJ disorders in the future.